So we have a couple of options, and I'm trying to think what the best way to do that is going to be. It says 9 minutes, so we may turn off it immediately at that point. I did want to at least go over the online questions that we had from last time. I'm still trying to figure out how to get D2L to talk to you and me and do all that stuff in an appropriate way. So you probably have all noticed, nobody got grades for any of them. Yes, and you're wondering what the heck was he asking? So we'll go over that pretty quickly. And then we'll do our clicker questions for today. And at that point, 8 minutes will probably be up and the projector will go off. At which point, what I was thinking about doing, I've got the presentation here. I may just go ahead and record it and put it online so you can take a look at it there, since I know a lot of you do that anyway. So is that going to work for people? OK, so again, and as you may guess, replacing the lamp on that is not straightforward. So let's move on, talk about the quiz questions that we had last time. Well, Friday during the day. Which of the following techniques would you use to determine one exact sequence of DNA that your favorite protein binds to in vitro? EMSA, DNA footprinting, CellX, chromatin IP, and DNA affinity chromatography. So DNA affinity chromatography depends on what? Depends on actually knowing the exact sequence that your protein binds to. So you're not going to use that to try and determine the exact sequence. OK, trying to determine the exact sequence, that's probably the most important thing here, is the one exact sequence. In vitro is going to rule out what? Rules out chromatin IP. Chromatin immunoprecipitation is in vivo, not in vitro. OK, so now we're down to EMSA, DNA footprinting, and CellX. EMSA is one piece of DNA, but you don't know exactly where your sequence is on there that your protein is binding to, so it's not EMSA. And that brings us to DNA footprinting and CellX. There may have been some confusion when I talked about CellX. At the end of CellX, you have a population of sequences. You have lots of different sequences that you can get a consensus sequence from, and then you can hopefully back-check with something like DNA footprinting to figure out exactly what that sequence is. So the correct answer here is DNA footprinting. The next of those questions, which, again, I apologize that these were all incorrect in the notes. I'll post the correct notes later today. I just noticed that right now as the other technical problems are happening. So which of the following proteins is one of those? OK, that's not very nice for terminology. Which, when expressed in fibroblasts, can convert those cells to pluripotent stem cells, LEX-A, NANOG, GAL4, mediator, TF2B. Well, that one hopefully everybody got right, because we've been beating on it a lot. It is NANOG. But what do we talk about in terms of LEX-A and GAL4? What's LEX-A? So LEX-A, we only really talked about as a DNA binding domain. And so it's a DNA binding domain from a bacterial transcriptional regulator. What about GAL4? GAL4 is a yeast transcriptional activator. So it has a DNA binding domain and an activation domain. And it was the experiment where they took the DNA binding domain from LEX-A and switched it with GAL4, which is where we were able to show that activation domains and DNA binding domains were really independent from each other. And it's just getting that specific activation sequence to the right place on the DNA, which is really critical for getting activation. Mediator is this humongous complex of proteins that's in between most of the transcriptional activators and all of your basal transcription machinery. But that's not true in all cases. You certainly have some gene regulatory proteins that interact directly either with the RNA polymerase or some of the other general transcription factors. So it's not always via the mediator. And TF2B does what? Binds to DNA at the TF2B recognition element at core promoters. So we've got 4 minutes for our clicker questions, which is almost exactly right. So let's go ahead and do those. Proteins that provide negative regulation of bacterial genes are most likely to bind downstream of the transcription start site, overlapping with the title box, RNA as it's being transcribed between the -10 and -35, upstream of -35, and is this actually going to start? Hopefully, yes, it is. Okay, good. So we are now counting. And if you think it didn't go the first time, hit it again. You can vote as many times as you like. This is not like normal elections, at least in this country. Okay, 30 and 2 minutes.
Oh no, that's just like jump. We lost like
This one's worse, yeah. Ten, 5, 4, 3, 2, 1. Okay, I'm going to do this really quickly because, again, we're just about to die in terms of the projector here. So most of you have it right. Yes, it's this one here between -10 and -35. That's where your negative regulators are most likely to bind in most cases. So our next clicker question, we'll start it now because we're just about to die. The tryptophan operon is generally thought to be an example of positive regulation, negative regulation, multi-factor regulation, chromatin remodeling, combinatorial control. Then I probably might have been able to get my whole lecture done in 9 minutes. This will die in just a second. And I'm curious if it's going to count down the seconds. We'll see. 30 seconds for the clickers anyway. So, boom. Okay, good. So we're gone. Still have another 20 seconds or so to vote for your clicker questions. Positive regulation, negative regulation, multifactorial regulation, chromatin remodeling, combinatorial control. Any binoculars in the back? So everybody done? Well, we've got 10 seconds left to do this. So, and I'll go back and talk about that previous question in just a second here. So we can go over it in a little bit more detail. Yeah. Pardon? Of course not. They don't trust me with it. Yeah, well, I know, but I should hack it. That would be a much better way to go, right? So, yes, it's tryptophan operon is a classic example of negative regulation. But hopefully what you got from Eric Iverson's lecture is that negative regulation is the binding of a regulator to the DNA and blocking the association of the RNA polymerase. And so that's the key also to the first question in terms of the regulatory aspects there. So that's the deal here. Before everybody disappears, so now we're going to continue with Lecture 20 after all the technical difficulties. Just a quick outline of what I'm going to talk about for the next 50 minutes or so. First, we'll start out by talking a little bit about DNA looping. Then, most of the time, we're going to talk about chromatin, exactly what you have to deal with when you're looking at the eukaryotic regulatory proteins and how those eukaryotic regulatory proteins
have to be used for that process to work properly in gene regulation. And then we'll move into a couple of regulation examples, and I will set my stopwatch to make sure that I don't go far too long because I have too many slides here. So that being said, let's start out talking about DNA looping. DNA looping has to do with how enhancers can act on proteins that are bound at promoters. And one of the really neat things that was found out about enhancers, now an enhancer is a sequence of DNA which binds to an enhancer binding protein. And those enhancers are really interesting because they can actually be present tens of thousands of base pairs away from the promoter. They can be present upstream or downstream from the promoter, and they can even be present on completely different DNA molecules. So that's just to say that you can have the binding site for a gene regulatory protein that's on a different DNA molecule than your promoter sequence that it's going to be interacting with, and this still functions. The way that people showed this was they had catenanes. These are two DNA molecules, circular DNAs, that are attached to each other with a sort of chainmail-like approach. So two rings that are linked to each other. And that was the way that we actually showed that these enhancer binding proteins were actually interacting with things at the promoter through a space interaction. So they were reaching out through space. It wasn't that the enhancer binding protein was scanning along the DNA, but it was literally looping. And that's what you can see up at the top of this figure here. Now, one of the things that's interesting about DNA looping, we've talked about DNA before and say that DNA is pretty flexible, but it turns out that that flexibility is relatively limited. So if you have two sequences and proteins binding those two sequences that are relatively close together, it turns out at about a certain point, and that's actually less than about 500 base pairs apart, it's actually kind of harder for these proteins to interact with each other because the DNA can't bend as easily. If you think about it, so DNA is a pretty bendy rod, but it's not completely flexible. So the closer you get, it's actually harder to bend. And so if you look here down at the bottom at the relative amounts of different proteins next to each other, depending on how far away they are from each other, there's a sweet spot at about 500 nucleotides away from each other. So how do we know all about this? Well, the main way that this is known is actually in looking at literally loops that form in the DNA while you have an enhancer binding protein, i.e. something that binds to DNA up to thousands, again, or 10,000s of nucleotides away, and then the RNA polymerase bound at a promoter. This particular example here is looking at the bacterial enhancer binding protein, NTRC, and a specific RNA polymerase. This is an RNA polymerase bound to one of the alternative sigma factors that you heard about from Eric Iverson the other day. And that polymerase with that sigma factor is really quite different than the other RNA polymerases bound with other sigma factors because it will bind to the promoter and sit at the promoter in a closed complex. So the DNA strands are both stuck together. This polymerase will not elongate unless there's an interaction between this particular RNA polymerase with that sigma factor associated with it and the enhancer binding protein. Now, this enhancer binding protein is actually shown here as a little lump in your textbook. You notice I've added a couple of extra balls to it because during my PhD, I actually showed that NTRC was present at the enhancer as a multimer. The other thing, and in fact the major part of my PhD thesis, was that the interaction between these two, this middle step here, the looped activation intermediate, is actually only happens very, very briefly during that process. There's also ATP hydrolysis. ATP hydrolysis, it turns out, takes place by the enhancer binding protein, and that's critical for getting open complex formation. But probably the most important, yeah, sure, fine, this was my PhD thesis, but the thing that really convinced people that the loops were what was going on is what's shown over here on the right-hand side. And this is work, again, done in the lab that I got my PhD in. It was published the year that I started my PhD, which is telling you how long ago that was. But here you can see two blobs, sort of a gray blob and a black blob. Well, the gray blob is the RNA polymerase, and the black blob is the enhancer binding protein. You can see very clearly that these two are associated with each other, and there's a loop of DNA in between. And so this, as it turns out, is the way that most enhancer binding proteins are going to be interacting with things at the promoter. And it's great to have a bacterial system because it's much simpler than the eukaryotic system. So that eukaryotic system, yes, it's mostly things interacting at a distance, but most of these proteins are not anywhere near as simple as the bacterial ones. There are lots of different proteins that are involved in transcriptional regulation, usually multiple proteins at any given promoter for any given gene. Not only are these DNA binding proteins, like this semicircular protein here, and then the various different proteins that are associated with it, all of these guys here. I can get my cursor to come onto the screen, that would be good. Oh, it's even a big one now too. Okay, so we have our partially circular protein binding to the DNA, and then various other different DNA binding proteins that are associated with it. But it's not only the DNA binding proteins that are involved in gene regulation. There also are many so-called co-activators or co-repressors, which you find in these systems. The big deal with a co-activator or co-repressor is these are non-DNA binding proteins, but still are very important for regulation of transcription. So, not surprisingly, a co-activator helps activate transcription, and a co-repressor helps to repress transcription. What's another aspect here which is important is that all of these interactions between the co-activators, the co-activator in and of itself could activate transcription just fine, but it needs to be brought to the right place. And bringing it to the right place is through these protein-protein interactions that happen with any of these DNA binding proteins which are present down here on the DNA. So a couple of aspects here. One, you notice that there are multiple different co-activators that could be interacting with different DNA binding proteins, just shown here as a cartoon, but there are many examples of how this works otherwise. But you can have then different DNA binding proteins, so here are the light blue and the green, or the purple and the green, which now interact with different co-activators. But you can also have co-repressors that could still be interacting with the same DNA binding protein over here that's important for interacting with co-activators can also be interacting with co-repressors. And so what that tells you is there's a whole mixture of different proteins that can be involved in this regulation process. You can have many, many different proteins all interacting with co-activators and co-repressors, which basically means the whole thing is really ridiculously complicated in terms of how it all fits together. So when people often look at this, what they do is they talk about something called the enhancesome, or for that matter also the repressosome, where you have multiple DNA binding proteins, and this is just shown here as cartoons. Here, this green one, the purple one, and the blue one are DNA binding proteins, as is this green and this, I guess, sort of brownish one over here. But you also have non-DNA binding proteins, and in some cases you even have some proteins which don't bind to DNA specifically, but their role serves to change the structure of the DNA in order to allow all of these DNA binding proteins and the other proteins that they associate with to then activate transcription. So multiple DNA binding proteins, multiple specific DNA binding proteins, very often non-specific DNA bending proteins, but then you also have these co-activators or co-repressors. So that's what you've assembled. If you look at this, it's actually a really pretty big set of different proteins that you have to get onto the DNA. Well, that brings us to eukaryotic DNA. Eukaryotic DNA is not a nice, simple gray line here. It's, of course, associated with lots of nucleosomes. And so that's just shown here as a cartoon. You can have nucleosomes around the transcription start site, which is shown here as TSS. You also, of course, have lots of nucleosomes which are bound to the DNA which is getting transcribed. That's shown as these little green balls here with tails. I give you one guess what those tails are for. Yes, they're histone tails, which then get modified. And it turns out that when you have transcription taking place, you have histone modifying enzymes, which not important what the names are here, but as you have transcription, the nucleosomes which are in front of the polymerase have one set of modifications, but then as soon as the polymerase goes by, you end up getting multiple other modifications which take place. And those are particularly at the fourth lysine, or the lysine, I should say, at the fourth position of the amino acid chain on histone H3, which becomes methylated. The more methylations it gets, mono, di, and tri, you can just see these by little purple flags here, 1, 2, and 3 little purple flags. This is basically a signal that this gene has just been transcribed. These are all bound to this little red blob here, and I give you one guess what that little red blob is. Of course, it's the C-terminal domain of RNA polymerase II. So that's what happens during polymerization. But what about setting stuff up in the first place? If you look at a typical piece of DNA, this is from a review article in Nature a couple of years ago now, but just look at where these nucleosomes are bound. And you see that the DNA is heavily wrapped around them. Now if you look at where activators are going to bind, that's what this ACT is here. This would be your gene regulatory protein. You've got a couple of possibilities. One, you have the binding site, shown here as BS, nice term, for binding to the activator. If it so happens that this binding site is somewhere where no nucleosomes are bound, and the reason that none of these nucleosomes are bound there is because they're specifically different nucleosomes. This is H2AZ. This is a nucleosome which has a alternative histone which has been associated with it. If these guys are bound to DNA on either side of this binding site, you're going to have a lot of binding of this activator protein to this binding site, which can then lead to assembly of the rest of the pre-initiation complex. On the other hand, many binding sites are often in what are these called covered promoters. And all that a covered promoter means is that that's a promoter where binding sites of one or many of the binding sites for an activator protein are blocked because there's nucleosome binding to these particular binding sites. So what you have to have take place here is that these nucleosomes have to move away so that the activator can bind to this. And in many cases, these are going to be promoters that have TATA boxes that are associated with them, bound to nucleosomes in such a way that you can't have the association, the pre-initiation complex, unless you have binding of these gene regulatory proteins, again here just shown as activators, and then remodeling of the chromatin in order to allow this TATA box to become available. Cairns, in this article in 2009, proposed a number of different ways to talk about open versus covered promoters, but the main thing here is that the open promoters, those that have binding sites for activators that are always available, very often have lots of A's and T's, a lot of times don't have TATA boxes, and usually have these variant histones. Not surprisingly, many of these are the essential genes. These are the genes which you need all the time. These are genes which are being transcribed all the time. And this is also a reminder that many of these promoters actually don't have Tata boxes, but they seem to be able to function quite well. On the other hand, the covered promoters, lots of binding sites for transcription factors, often have Tata boxes, don't have these variant histones, lots of change in histones, and clearly these guys are much more of the regulated genes which you find there. So let's look at this in a more general sense. This is the figure from your textbook, where you have the case of binding of a gene regulatory protein, our little pawns here from chess, our gene activator protein, but the Tata box, which is buried in chromatin. Couple of things can happen here. Most of them depend on the presence of a chromatin remodeling complex. Chromatin remodeling complexes, of course, require ATP in order to move nucleosomes around, which you have here, and often once these nucleosomes have been moved around, histone chaperones will either completely remove the histones, allowing access to the Tata box, or put in some of these alternative histones into the nucleosome, again, opening up this Tata box appropriately. A third process, which actually doesn't need this way, maybe you should call it a fourth process, that doesn't require this chromatin remodeling complex, is just binding to a histone modifying enzyme. And in the case of a gene activator protein, this would normally be a histone acetyltransferase, which will now put acetyl groups onto those otherwise positively charged side chains that you have on the histones. And in that process, lead to opening and accessibility of the Tata box. Once you have accessibility of the Tata box, now you can add all of your general transcription factors, of course starting with TF2D, which binds to the Tata box, TF2B, TF2E, TF2F, TF2H, etc., your mediator complex and your RNA polymerase, so now you can finally get transcription to take place. In the same modeling system here, where you talk about looking at what happens, where you have either your binding site here, or you can also think of this as the Tata box. Activator protein can't associate with the DNA once it has one of these chromatin remodeling complexes. ISWE and Remodel are both chromatin remodeling complexes. Can't bind. A different chromatin remodeling complex, the SNF complex, which we've talked about before, can either move the nucleosomes along so that they're not on this binding site anymore. They can completely remove one of the nucleosomes, allowing access to this site. Or, in some cases, again, this is now our histone chaperone here, which will change out the histones here and then allow the association of your activator binding protein to this particular site. So what do these chromatin modifications do for us, or particularly modifications to the side chains? These again could either be an alternative histone, or more likely it's some kind of histone modification enzyme which has been brought to this region of the DNA by interactions with a regulatory protein. So many of these regulatory proteins, as we talked about before, interact with co-activators or co-repressors. Well, what do co-activators and co-repressors often do? Well, they'll either interact with chromatin remodeling complexes, or in many cases, they will also interact with histone modification enzymes. So those histone modification enzymes very often are going to be something like a histone acetyltransferase. So here's a nice example of that. Here we have our happy chromatin with its nucleosome and all of those histone side chains sticking out. Gene activator protein, which binds to DNA somewhere, potentially long ways away. This is our enhancer. Then it interacts with a co-activator. This co-activator has histone acetyltransferase activity that will acetylate particular histone in histone H3. Often these interactions will lead to more interactions. You remember in the histone code, we often have multiple different modifications that happen at the tails of the histones. Here we have a histone kinase, which associates with histone H3, now phosphorylates at position serine 10. Now you've got acetylated lysine K9, phosphorylated serine 10, again, either the same or a different histone acetyltransferase will modify lysine at position 14, also add a acetyl group at this position. Together with the chromatin remodeling complex, this will move the histone along, but at the same time, it turns out some of these modified side chains actually interact with some of the subunits of TF2D. Well, this makes perfect sense. You want to have TF2D because one of the other subunits of TF2D is the TATA binding protein. So TF2D you can actually bring to place near a promoter by it binding to these modified histone side chains. Once you have TF2D that's assembled, then of course you're going to get TF2B and all the rest of your transcription pre-initiation complex, which eventually leads to transcription. So that's the case for one transcriptional activation protein. Well, one of the interesting things that was found very early when people were studying eukaryotic transcriptional regulation is what I've rather tongue-in-cheek cartooned here, is that 1 + 2 = 100. And all that that means is when you have two regulatory proteins, particularly if you've got two gene activator proteins, that interact with the regulatory region of a gene, one of these will stimulate transcription a little bit, the other one will stimulate transcription a little bit, but the two of them together are way more than the sum of these individual ones here. And this is really what you see in terms of eukaryotic gene regulation. You have multiple proteins, and those proteins together are much more important and much more active than you would have with just a single one of these proteins alone. So let's look at this overall activation process. This is a cartoon of what could happen at one particular case, and it turns out this is slightly different for all kinds of different genes. So one gene may have one of these steps, another gene may have a difference of these steps, but if you had all of these together, this is going to be the vast majority of the kinds of gene regulation that you have. So, first step, your activator protein binds to chromatin. And so chromatin, of course, this is our DNA, which is associated with histones in the case of a eukaryotic gene. So this gene activator protein binds to chromatin. Usually it's going to be binding to an enhancer, which again can be thousands of base pairs upstream, or for that matter downstream of the promoter, which is going to be regulated. That very often associates with the chromatin remodeling complex, and again, many of these chromatin remodeling complexes are also going to be co-activators. You have chromatin remodeling, moving histones around back and forth, sometimes even taking them out completely. Then you'll have the interaction with histone modification enzymes. Again, these are often co-activator proteins, which will then associate with first your activator proteins, which have associated with the DNA, and then will modify the histones nearby. This often is going to allow other activator proteins to be able to associate with the DNA, and hopefully you can sort of see this as a kind of a little snowball. You get the little snowball, starts rolling, gets more and more and more. bigger and bigger and bigger, more and more proteins, and usually here less and less DNA, which is associated with these nucleosomes. So after these other activator proteins are bound, now usually multiple activators, multiple co-activators associated with the DNA will now bring the general transcription factors in the RNA polymerase to the promoter. So you've got the mediator complex, these general transcription factors, the RNA polymerase, put together the pre-initiation complex of 100-odd proteins present at the promoter, which is ready to go, but usually requires yet another little kick, either in terms of releasing this protein from, or say the protein complex, I should say, from a pause near to the transcriptional start, or stimulating the activity of TF2H, so you get an open complex formation. And very often these will be a rearrangement of the proteins, moving them around in the actual complex until you get an open complex, and eventually an elongating RNA polymerase. So far we've only talked about activators. We also need to talk about repressors. There are repressors in eukaryotic transcriptional regulation which function very similar to what's going on with bacterial repressors. These bacterial repressors, of course, bind to the DNA, and a bacterial repressor is usually going to be blocking the binding of RNA polymerase. Here, there are some cases in eukaryotes where the repressor blocks the binding of the RNA polymerase, but usually they block the binding of an activator protein. So here, as we can see up at the top, we've got a repressor protein in red, the activator protein in green. The repressor protein binds to DNA, which overlaps with the DNA sequence that the activator protein would bind to, so that this activator protein can't bind to DNA, or at least it doesn't bind DNA as well as it would in the absence of the repressor protein. But since in the case of eukaryotic regulation, very often there are interactions at a distance, either with the proteins that are present at the promoter or with other proteins, and the activation domain of a lot of these proteins is basically really kind of sticky and kind of independent of the DNA binding domain. Many repressors actually act by sticking to that sticky part and not allowing it to interact with all of the other transcriptional regulators that it would need. So we've got either a direct competition for DNA binding, binding to a enhancer binding protein and repressing this, or in a few cases you actually have a direct interaction with some of the general transcription factors. So here, binding your repressor binds to DNA, interacts with some of the general transcription factors in the same place that an activator otherwise would be interacting with them. And this interaction actually blocks the transcription from taking place. Usually this would be in one of those stalled promoter complexes where the polymerase is just waiting to move forward. Here it's blocking that. Or you could also block the assembly of the rest of the pre-initiation complex. If you would bind to a surface of TF2D that would then block the binding of TF2B, you're not going to get a fully active process. These are also called direct mechanisms for having repression. There are also a number of indirect mechanisms which function not unlike what you have with transcriptional activators, which bind a long way away from the promoter. These repressors that act indirectly can also bind to chromatin remodeling complexes, but instead of that chromatin remodeling complex opening up the DNA, it now closes it down and removes the Tata box. These repressor proteins often interact with either histone deacetylases or histone methyltransferases, Histone deacetylases will, of course, take off the acetyl groups that were added to the histones by the histone acetyltransferases, and thereby restoring the positive charge which these have, allowing them to bind better to DNA. Or in many cases, you also have co-repressor proteins, which are histone methyltransferases. These will phosphorylate, they will methylate the side chains of each of these histones, and that methylation will usually lead to binding of other proteins that will associate with methylated histones. This often leads to a pretty strong heterochromatin formation. So methylation of histone side chains very often will lead to this kind of repression. This, of course, is indirect repression because there's no interaction here between the repressor and the actual Tata box. It's all an indirect process, usually mediated through nucleosomes. So what do these things actually look like? Here's an example of two, in fact, enhancesomes at a high-resolution crystal structure. Here we have three different transcriptional activation proteins, BATF and June. Again, the names here are not critical. And IRF4, these interacting with a particular DNA binding site, leading to activation of certain genes. Turns out that the same proteins, again, IRF4, BATF, and June, can interact with a different DNA binding site. And it turns out it's just because it's flipped over onto the opposite strand here. And that interaction now leads to the same kind of differentiation and activation of these different genes. So cooperative binding of these proteins to each other, which leads to the first part of binding to one of these enhancesome processes. So how are we doing for time here? We've already had about half an hour. Why don't we go through one or two of these examples, and then we'll call it a day as far as lecturing is concerned. You haven't been able to slow me down today because I've just been talking to my computer as opposed to actually really properly lecturing here. So we'll start out talking about positional expression in Drosophila embryos. So why should we do this? Well, mostly because Drosophila has turned out to be an extremely good model organism for understanding gene regulation, and particularly combinatorial gene regulation, as we'll see in the next couple of slides here. Drosophila, as any of you know who have compost buckets, are fruit flies, and fruit flies do extremely well in climates such as Portland, and you have lots and lots of these embryos. At least my compost bucket has lots and lots of these embryos in them. So they're really very easy to study, and it turns out that you can also quite easily make mutations in these Drosophila. The original ones were just made by X-raying the fruit flies, and so original genetics. In fact, even before people knew that DNA was genetic material, they were X-raying fruit flies and taking a look at them. And really, it was the pioneering work of Christine Nusslein-Volhard and her group at the Max Planck Institute for Developmental Biology in Tübingen, which really established the fruit fly as a model organism for looking at development and how embryonic genes get turned on. Those of you who are in developmental biology, hopefully this will all be review for you. But what we're interested in here is looking at how gene expression in different parts of the embryo leads to expression of other genes. So basically, those are gene regulatory proteins leading to the expression of other gene regulatory proteins, and how does that lead to specific expression inside your Drosophila embryo. One of the things that we're looking at here is a typical Drosophila embryo, at least in cartoon form. All the little circles represent nuclei, and they're basically one big cell. This is what I say here by syncytia. And because it's one big cell, any particular protein which is made can diffuse to near where it's actually being made. And so if you look at the bicoid gene and the hunchback gene, actually the bicoid gene originally comes from the mother. This is present at a gradient starting at the anterior end of the embryo. And as they grade in, get into smaller and smaller amounts as it moves towards the posterior. Same thing is true of this protein hunchback. Again, the Drosophila geneticists get to name all of these cool names, basically based on what they saw when they had a mutation in this particular gene. So a mutation in this particular gene leads to a hunchback phenotype. Mutation in this particular gene here, which is expressed in these two areas in the embryo, is called giant. So if you don't have this gene, then the embryo is much, much larger. And this one down here, Krippel, again, they were mostly German researchers who found this in the first place. This looked like a crippled embryo. How you know an embryo is crippled, I don't know, but these are the names which were given to you. Each of these represents a single gene regulatory protein. So these gene regulatory proteins lead to the expression of a whole set of different genes. But we're going to look just at one, and this is called the EVE gene, or even-skipped gene. Here, the even-skip gene is shown here in various different stripes in the embryo. You look here, there are actually 7 different stripes. Green here, where they're not overlapping with expression of the giant protein. Yellow, where of course red and green come together. So 7 different stripes, 1, 2, 3, 4, 5, 6, 7 stripes of even skipped. And that's because each of the segments that you have there. And then the giant protein, as I showed you before, much larger bands here, both of them in red. And what's really amazing is the expression of just these 4 gene regulatory proteins that leads to expression of even skipped in all of these different regions of the embryo. How does that work? Well, how do we figure it out? Basically, it was by looking at the genes that are present in the Drosophila embryo, and particularly the regulatory sequences which are upstream of this even skip gene. It's got a Tata box, a normal start of transcription, and then multiple different regulatory elements, all of them present upstream here of the Tata box. And it turns out that of these 7 stripes that you have here, 1, 2, 3, 4, 5, 6, 7, each, slight exception, but basically each of these individual segments turns out to be important for regulation of even skipped in one of these individual segments. How do we know that? Because if you take one of these regulatory segments, take it out of the normal gene expression pattern here, say the normal gene, put it into a reporter gene, which is now expressing a gene that you can notice the activity of. In this case, it's LAC-Z, which is the bacterial beta-galactosidase gene, which if you incubate it together the appropriate dye will turn blue. Just take this one piece of the regulatory DNA, which normally all of these segments would lead to 7 stripes. One segment leads to expression in one and only one stripe in the Drosophila embryo. Turns out that this is about a 20,000 base pair regulatory region. What's present in that regulatory region? What's present in the DNA? Well, it turns out, not surprisingly, that this piece of DNA has a bunch of potential binding sites. And so when you say potential binding sites here, this gets back to some of the experiments which we talked about last time, or at least in my last lecture, where we looked at band shift assays, for instance. If you took this piece of DNA, the stripe 2 regulatory region, this module here, 480 of that about 20,000 total that we had on the last slide. So the 20,000 here, that's about this piece here. The regulatory region is about 20,000. You take about 500 bases of that, and that's this piece. So you could use this in an electrophoretic mobility shift assay. What you see is that there are multiple different proteins, and here 4 particular ones, which are going to bind to this particular piece of DNA. You go and look in a little bit more detail, again by doing DNase footprinting or some kind of immunoprecipitation. What you see is there are multiple possible binding sites for cripple, bicoid, hunchback, and giant. Those are shown here, little cartoons up at the top. Hunchback is a little mushroom, bicoid is this little curve here, cripple is this sort of small hat, and giant is kind of a big hat. What we see is that when you have binding by the cripple or giant protein, these repress gene expression. Bicoid and hunchback, on the other hand, will activate. And so this brings us to basically what we're looking at here. Here again, as I showed you before, bicoid is present in a gradient from one end of your embryo. Hunchback is present in a gradient from one end of the embryo. Giant is present in these 2 pretty broad stripes in the embryo. And cripple is present at one other side of the embryo. So these are all the proteins that could bind. And again, this is stuff which was shown through things like these band shift assays, etc., with purified proteins. But what's going on here in stripe 2, where even skipped is normally being expressed? What happens is, it turns out that this region of the embryo, right here, is where you have no giant being expressed. And that's here. It's this posterior end of where giant is being expressed. And before you have the expression of cripple. So basically it's right in this region right here, but you still have this activation of transcription because you've got 2 positive regulators, hunchback and bicoid, which are still present in this particular region of the genome. So here you have combination of different regulatory proteins, 2 positive regulators, 2 negative regulators, leading to expression of your protein in one particular and actually very specific place in the embryo. If you go anywhere anterior to this, you're not going to get expression of this gene because the giant protein is around. This is repressing transcription. If you go anywhere posterior, you're not going to get expression of this gene because the cripple gene is there. If you go too far posterior, you don't have any of the hunchback and bicoid gene either, and so anywhere which is going to be posterior of cripple is not going to be expressing it. So now, this combination of genes that we had here, this combination of DNA binding sites, I should say, leads to expression of even skipped in this stripe of the embryo, and only in this stripe of the embryo, because everywhere else, at least given the gene regulatory proteins here, is going to lead to expression just in this one place. So that's even skipped. That's one way that we can be regulating the activity of these particular genes. There are other ways to do it, just the presence and absence, which is just binding. There are other possibilities as well. Basically, this is how you get your transcriptional activator in the first place, just by making it. Protein synthesis, you don't have it, then you'll have it. Many times you're going to have ligand binding, and we'll look at some examples of this, again, probably next lecture at this rate. Ligand binding, which will lead to activation of an already present protein. And as you remember, this is actually really quite a fast way to get your gene expressed in the first place. Covalent modification, phosphorylation in particular. A few cases, you have a separate subunit that's going to associate with your otherwise active DNA bindings or active transcriptional activator. Can't do anything until it's bound to DNA, so it has to have a DNA binding subunit. number of cases. You've got DNA binding proteins that are transcriptional activators that can only be active if you remove a DNA binding protein. Sometimes that removal will allow that protein to go into the nucleus. Remember, all of these transcriptional activators have to be present in the nucleus in order to have their full activity. Or in a few cases, you actually have proteolytic cleavage, which leads to the activation of a gene regulatory protein by going through the membrane. So I think at this point, given the time, we'll stop and we'll talk about how the expression state of genes is being regulated on Wednesday, and hopefully our projector will actually be working.
I did want to ask what you thought about the whole online quiz thing versus being forced to be here to actually get the 2 measly points that you would get for a particular lecture. So do you have comments on that, or just, yeah?
I like it as an option, so having both, if it works logistically, I think is good.
Yeah, so we've got a vote, 1 of 150, for having both as an option. And second, okay, we've got 2 votes. Yeah.
I think, at least on Friday, I would have liked it better if we would have been able to go back and look through the quiz after we had submitted it, and it would have shown us this answer was correct or this answer was incorrect, even if we didn't have an explanation.
Yeah. So knowing, having some kind of feedback would be really useful, and I totally agree. I'm trying to do that. I'm wrestling with D2L to try and do that. But I'm definitely planning on having that, and so you'd actually have complete feedback. Once you've submitted your answers, you should be able to get the proper answer to that. Don't tell anybody who hasn't taken it yet. But yeah, that's one of the reasons to maybe leave that a little bit or have it the next day after it's expired. So we're still trying to deal with that. Yeah, other comments?
I was going to say, like, I've taken online classes on D2L before, and I don't like that it doesn't prompt, like, it doesn't remind you, because I totally forgot about them, because I'm not used to doing clickers online.
Okay.
It's fine, it'll normalize, but usually, like, when you take an online class, it'll prompt you in your news feed or something, or up in the top, it'll be like, you have a quiz coming up.
Yep. So that's a great idea. Now I'm just repeating this because it's going to be taped. So yeah, having some reminder. Hey, by the way, you have a quiz that you haven't taken yet. Might be a really good idea to do so. Something like that. And again, I'll tweak and I'll see if I can figure that out. Those of you who are lucky enough to take virology next term, be warned. I'm going to be taking some of these comments there. Yeah. Right. So whether it actually... Right, yeah. So the basic problem is you submit your answers and they disappear into the ether, and who knows? Al Gore is checking them. Yeah.
I think as an option would be probably the better, just because it's nice to have the collaboration with classmates here.
Right. So again, the question is to have it as an option. And if I'd actually had time this morning, if we hadn't had all these issues, and I'll probably actually put in a clicker question next time, assuming this is actually going to work, about what you think about it. And so we'll just have a poll and see what people think. It's a bit more work for me to do both simultaneously, but yeah, it might be worthwhile. Okay, but I'll do that. We'll do that with clickers next time rather than. So, we got any other questions, comments? Yeah.
What if you just had the quiz online and then talk about it in class next time?
Right. So that would be one of the options. So one of the options would be to have the quiz online and then, just like I did today, talk about the answers. Yeah, we'd gain 5 to 10 minutes lecture time. Is exactly right. Although discussing them, which would be, so that'd probably be about a balance between the amount of time that we would end up spending doing the clicker questions and talking about the clicker questions. So, yeah, I think taking that's sort of the point. It's kind of one of the things I like about doing the clicker questions here is that you actually have a bit of a group interaction, and so taking the online quizzes as a group, I know a few people who will remain nameless who have been getting together and doing the quizzes online together, at least talking about them. Before they actually do them, or have one person, guinea pig then go from there. So that I think is a great idea. And part of the reason that, again, I do clickers is to have that sort of group interaction. So there may be a way to do that on D2L as well, then have a consensus vote. That would be really cool if we could. Again, I'm still looking into that. Okay, other questions, comments, etc. I will post this lecture online this afternoon. And there will be clicker questions on what you think about clicker questions and online questions on Wednesday. And we'll also have clicker questions on the material from today on Wednesday. Yeah.